Best Linear Unbiased Prediction, often known as BLUP, is a statistical methodology widely applied in genetic evaluation, particularly in animal and plant breeding. The method is crucial in genomic selection, where breeders aim to predict the genetic potential of an individual based on genetic markers, rather than just phenotypic observations. Developed by Charles Roy Henderson in the 1950s, BLUP has transformed how genetic values are estimated by effectively accounting for environmental effects and improving prediction accuracy. This statistical tool remains the gold standard for estimating breeding values in complex genetic models. Genomic selection, an advancement built upon traditional BLUP, incorporates genome-wide markers to predict breeding values, thus enabling the early selection of superior candidates. This technique has revolutionized breeding programs by increasing selection efficiency, reducing generation intervals, and enhancing genetic gain. The integration of BLUP with genomic data has resulted in Genomic Best Linear Unbiased Prediction, or GBLUP, which is widely used in both livestock and crop improvement programs. By considering entire genomes rather than a subset of genetic markers, BLUP-based genomic selection minimizes biases and maximizes accuracy in breeding value predictions. Best Linear Unbiased Prediction, or BLUP, is rooted in statistical genetics and linear mixed models, making it one of the most robust methods for estimating genetic values in both plant and animal breeding. The foundation of BLUP is based on the principles of least squares regression, variance components, and random effects modeling. It was first formalized by Charles Roy Henderson, who developed a series of equations known as Henderson's mixed model equations, or MME. These equations enable the estimation of fixed and random effects while minimizing bias and maximizing prediction accuracy. The statistical rigor of BLUP ensures that it provides the most accurate and reliable genetic evaluations, even in complex breeding scenarios. BLUP operates within the framework of a linear mixed model, which accounts for both fixed effects, non-genetic factors, and random effects, genetic contributions. The general form of the model can be expressed as y equals xb plus zoo plus e, where y is the vector of observed phenotypic values such as milk yield, plant height, or disease resistance scores. xb represents the fixed effects, which may include environmental factors like temperature, nutrition, management practices, or location. These effects are assumed to be known or estimated with negligible error. Zoo represents the random genetic effects, capturing the breeding values of individuals. These effects are considered random variables with a mean of 0 and a variance that depends on the genetic relatedness of individuals. Finally, E is the residual error, accounting for unexplained variation due to measurement errors, unknown environmental effects, or other sources of randomness. This formulation allows breeders to separate genetic contributions from environmental influences, improving the accuracy of selection decisions. The name Best Linear Unbiased Prediction comes from the statistical properties that make BLUP an ideal method for genetic evaluations. Each component of the name signifies a specific advantage, best. Among all possible linear predictors, BLUP minimizes the prediction error variance. This ensures that the estimated breeding values, or EBVs, are as close as possible to the true genetic values. Linear BLUP relies on a linear model where predictions are made as linear functions of the observed data. This makes computation efficient and ensures compatibility with standard statistical methods. Unbiased BLUP ensures that estimates do not systematically overestimate or underestimate genetic values. It accounts for the statistical distribution of genetic effects, preventing bias due to environmental factors or selection. Prediction. Unlike traditional estimators that simply describe past data, BLUP is predictive in nature. It forecasts genetic merit for future generations, making it highly effective for breeding programs. A key aspect of BLUP is its reliance on variance components, which quantify the proportion of phenotypic variation that can be attributed to genetic and environmental factors. These components are estimated using techniques such as restricted maximum likelihood, or REML, which ensures unbiased and efficient estimation. The main variance components in BLUP include genetic variance, which represents the contribution of genetic factors to phenotypic differences; residual variance, which accounts for environmental and other non-genetic sources of variation; heritability, defined as the proportion of total phenotypic variance attributable to genetic factors, computed as the ratio of genetic variance to the sum of genetic and residual variances. Heritability determines how effectively genetic selection can improve a trait. Henderson's mixed model equations provide the mathematical foundation for computing BLUP estimates. These equations are formulated as x transpose x, x transpose z, z transpose x, z transpose z plus lambda y times beta u equals x transpose y, z transpose y, where x transpose x and z transpose z are matrices of fixed and random effects, respectively. Lambda 1 is a penalty term that accounts for genetic variance and avoids overfitting. X transpose y and z transpose y represent observed data used to estimate effects. Solving these equations yields the estimated breeding values, or EBVs, for each individual. The efficiency of these computations depends on the size of the data set and the structure of the relationship matrix. While BLUP is widely used, several alternative methods exist for genomic selection. These include ordinary least squares, or OLS. Unlike BLUP, OLS assumes all effects are fixed and does not incorporate genetic relationships. This makes OLS less efficient for breeding applications. Then we have Bayesian regression methods like Bayes A, Bayes B, and Bayes C. These methods introduce prior distributions on genetic effects, allowing for non-uniform shrinkage. Bayesian approaches may outperform BLUP in cases where few major genes control the trait. And finally, there are machine learning models such as random forest, neural networks, and support vector machines. These models can capture nonlinear interactions between genetic markers, but they often require large data sets and are computationally intensive. Despite these alternatives, BLUP remains the preferred choice for most breeding programs due to its efficiency, statistical rigor, and ability to handle large-scale genomic data. Multiple studies have demonstrated the accuracy and reliability of BLUP-based genomic selection. In dairy cattle breeding, BLUP-based models have achieved prediction accuracies of 0.7 to 0.9, depending on trait heritability. In maize breeding, the use of GBLUP has resulted in a 15 to 20% improvement in genetic gain compared to traditional selection. A study by Moiwissen et al. In 2001, comparing BLUP with Bayesian methods, found that while Bayesian models provided slightly higher accuracy for some traits, BLUP remained the most practical and widely applicable approach due to its computational efficiency. Additionally, research on disease resistance in wheat using BLUP has shown that genomic selection can predict resistance scores with an accuracy of 0.6 to 0.8, significantly reducing the need for field trials. Despite its strengths, BLUP has some limitations. It assumes a linear relationship between genetic markers and phenotype, which may not fully capture epistatic or gene-gene interactions. Moreover, BLUP relies on estimated variance components, which may introduce uncertainty if sample sizes are small. To address these challenges, researchers are developing hybrid models that integrate BLUP with deep learning and nonlinear statistical techniques. The integration of multi-omics data, such as transcriptomics and proteomics, into BLUP models is also a promising direction, enabling more precise genetic predictions. As computational power increases, BLUP will continue to evolve, incorporating new data sources and methodologies to enhance its predictive power. The future of BLUP lies in combining traditional statistical approaches with machine learning, expanding its application beyond agriculture into personalized medicine and evolutionary biology. Best Linear Unbiased Prediction, or BLUP, has been a transformative statistical tool in the field of animal breeding, revolutionizing the way breeders estimate genetic merit and improve livestock populations. Since its development by C.R. Henderson in the mid-20th century, BLUP has provided a robust framework for genetic evaluation by accounting for both fixed and random effects in complex breeding populations. The application of BLUP in animal breeding has significantly improved the accuracy of estimated breeding values, or EBVs, allowing for more efficient selection of superior animals. By integrating pedigree and performance data, BLUP has become the gold standard in dairy cattle, beef cattle, swine, poultry, sheep, and aquaculture breeding. And dairy cattle breeding is one of the most well-documented success stories of BLUP implementation. Since the introduction of BLUP in the 1970s, genetic progress in milk production, fertility, disease resistance, and conformation traits has accelerated significantly. Before BLUP, dairy breeders relied on phenotypic selection, which was highly influenced by environmental factors. BLUP allowed for the separation of genetic and environmental components, leading to more accurate selection of high-performing bulls and cows. The most notable impact of BLUP in dairy breeding has been seen in milk yield improvement. In Holstein cattle, the breed that dominates global milk production, genetic progress has resulted in an increase of approximately 100 kilograms of milk per cow per year since the widespread adoption of BLUP. Studies conducted by the United States Department of Agriculture, or USDA, have shown that BLUP-based genetic selection has led to annual genetic gains of 1.5 to 2% per year in milk production. Another major success of BLUP in dairy breeding is its application in longevity and fertility traits. While early genetic selection focused heavily on milk yield, this led to unintended declines in fertility and productive lifespan. By incorporating BLUP models that estimate genetic merit for fertility-related traits such as calving interval, conception rate, and daughter pregnancy rate, breeders have reversed these negative trends. Genetic trends indicate that fertility rates, once declining in Holstein cattle, have now stabilized or even improved in some breeding programs due to BLUP-driven selection. BLUP has also played a key role in disease resistance and animal welfare. One of the most costly diseases in dairy cattle is mastitis, an inflammatory condition of the udder that affects milk quality and yield. Genetic evaluations using BLUP have identified bulls with improved mastitis resistance, leading to healthier herds and reduced veterinary costs. Similarly, selection for improved hoof health, somatic cell count, and metabolic stability has been enhanced using BLUP, contributing to overall herd well-being. The application of BLUP in beef cattle breeding has enabled significant genetic gains in growth performance, feed efficiency, and carcass quality. The beef industry relies on optimizing traits that enhance productivity while maintaining adaptability to different environments, and BLUP has been instrumental in achieving these goals. One of the most important traits in beef production is weaning weight, as it directly affects profitability. BLUP-based selection programs have consistently increased weaning weights in commercial beef herds with genetic trends, indicating annual improvements of 2 to 4 kilograms per year in some breeds, such as Angus and Hereford. Similarly, post-weaning growth rate and carcass weight have seen steady genetic progress due to the accurate estimation of breeding values using BLUP. Another key trait improved through BLUP is feed efficiency, often measured as residual feed intake, or RFI. Feed costs represent the largest expense in beef production, and selecting animals that can convert feed into body weight more efficiently is crucial for sustainability. BLUP has facilitated the selection of bulls with superior RFI values, reducing overall feed consumption while maintaining or increasing weight gain. Carcass quality traits, such as marbling, ribeye area, and fat thickness, have also been enhanced using BLUP. The integration of carcass ultrasound data into genetic evaluations has allowed breeders to make more informed selection decisions, leading to beef products with better marbling scores and improved consumer appeal. Beyond economic traits, BLUP has been utilized in environmental adaptability, particularly in selecting heat-tolerant cattle. In tropical and arid regions, heat stress reduces productivity and reproductive efficiency. By incorporating temperature-adapted performance records into BLUP models, breeders have identified genetic lines with improved thermotolerance, leading to cattle breeds that perform well in challenging climatic conditions. The pig industry has benefited immensely from BLUP-based genetic evaluations, particularly in improving growth rate, reproductive efficiency, and carcass traits. Swine breeding programs worldwide have implemented BLUP to enhance productivity while maintaining genetic diversity. One of the most important economic traits in pig production is growth rate, typically measured as average daily gain, or ADG. BLUP has allowed for consistent genetic progress in ADG, with studies reporting improvements of 10 to 15 grams per day per year in commercial pig lines. This has significantly reduced the time required to reach market weight, increasing profitability for producers. Reproductive traits in pigs have also been improved through BLUP, particularly litter size and sow longevity. Selection for number of piglets born alive, or NBA, has led to steady increases in litter size over the past 3 decades, with genetic gains of 0.1 to 0.2 piglets per litter per year. However, this has also introduced challenges such as increased piglet mortality. By incorporating maternal genetic effects into BLUP models, breeders have been able to balance litter size with improved piglet survival rates. Carcass quality is another area where BLUP has had a significant impact. The selection for lean meat percentage and back fat thickness has resulted in pigs with higher muscle content and reduced excess fat, improving both production efficiency and consumer preference. Genetic evaluations using BLUP have also led to improvements in meat tenderness and color stability, key factors in pork marketing. The poultry industry, particularly broilers or meat chickens and layers, which are egg-producing hens, has seen some of the most dramatic genetic improvements due to BLUP-based selection. The ability to rapidly generate new generations in poultry allows for accelerated genetic progress. In broiler breeding, BLUP has been used extensively to enhance growth rate, feed efficiency, and breast muscle yield. Over the past 50 years, the genetic gain achieved through BLUP has resulted in broilers that grow nearly 4 times faster than their ancestors, while consuming less feed. Current broiler strains can reach a market weight of 2.5 kilograms in just 35 to 40 days, compared to 16 weeks in the 1950s. In layer breeding, BLUP has been instrumental in improving egg production, eggshell quality, and disease resistance. Modern laying hens produce over 300 eggs per year, a significant increase from the 200 to 250 eggs per year seen before the introduction of BLUP in breeding programs. Additionally, selection for improved eggshell strength has reduced breakage rates, improving overall efficiency. BLUP has also been widely applied in sheep and goat breeding, with significant genetic improvements in growth rate, milk yield, fleece quality, and disease resistance. In meat sheep breeds, BLUP has led to improvements in weaning weight, post-weaning growth, and carcass yield. Genetic evaluations in Texel and Suffolk sheep have resulted in lambs that grow 15 to 20% faster than those selected without BLUP. In wool sheep breeds, BLUP has been instrumental in improving fiber diameter, staple length, and fleece weight. Merino breeding programs using BLUP have successfully produced sheep with finer wool and increased fleece yields, ensuring competitiveness in the global wool market. In dairy goat breeding, BLUP has significantly enhanced milk yield and protein content, making dairy goats a viable alternative to cows in certain regions. Genetic evaluations have identified superior bucks, and does that consistently produce high-quality milk with improved fat and protein composition. The application of BLUP in animal breeding has led to remarkable genetic progress across multiple livestock species. By providing accurate, unbiased estimates of genetic merit, BLUP has enhanced productivity, efficiency, and sustainability in dairy, beef, swine, poultry, sheep, and goat breeding. The integration of genomic data, or GBLUP, continues to improve the precision of genetic evaluations, ensuring that BLUP remains an indispensable tool in modern breeding programs. The application of Best Linear Unbiased Prediction, or BLUP, in genetic evaluations has been instrumental in enhancing disease resistance and environmental adaptation in both livestock and plant breeding programs. As global agriculture faces growing challenges due to emerging diseases, climate change, and environmental stressors, BLUP-based models have played a critical role in identifying and selecting resilient animals and crops. By leveraging large-scale genetic data, BLUP enables breeders to estimate genetic merit for resistance traits with high accuracy, leading to healthier populations and more sustainable production systems. One of the most significant applications of BLUP in breeding programs has been its role in selecting animals and plants with improved resistance to infectious diseases. Disease resistance traits are typically low to moderately heritable, with heritability ranging from 0.1 to 0.4, making their improvement challenging through traditional selection methods. However, BLUP allows for the estimation of genetic breeding values, or EBVs, for resistance traits, improving the efficiency of selection by separating genetic effects from environmental influences. In the dairy industry, mastitis is one of the most economically significant diseases, costing the global dairy sector billions of dollars annually due to reduced milk production, veterinary expenses, and discarded milk. Genetic evaluations using BLUP have enabled the identification of sires and cows with improved resistance to mastitis, leading to healthier herds. BLUP models incorporate somatic cell count, or SCC, as an indicator trait for mastitis resistance, as SCC is strongly correlated with udder infections. Studies have shown that genetic selection based on BLUP can reduce mastitis incidence by 20 to 40% over multiple generations while maintaining milk production efficiency. Similarly, BLUP has been used to improve resistance to bovine respiratory disease, or BRD, John's disease, and digital dermatitis. For instance, genetic evaluations for bovine tuberculosis resistance have been implemented in breeding programs in the United Kingdom and Ireland, where disease prevalence has been a persistent issue. In beef cattle breeding, BLUP has been effectively applied to enhance resistance to internal and external parasites, respiratory diseases, and metabolic disorders. One of the most impactful genetic evaluations has focused on bovine respiratory disease, or BRD, which affects feedlot cattle worldwide. By using BLUP-based models, researchers have identified genetic lines with improved immune response, resulting in reduced morbidity and mortality rates in feedlot operations. Additionally, BLUP has been utilized to select animals with resistance to ticks and gastrointestinal nematodes, or worms, which cause significant economic losses in tropical and subtropical beef production systems. A notable study in Brahman and Angus cattle found that genetic selection for parasite resistance using BLUP reduced worm burden by 30 to 50% over successive generations, reducing the need for chemical dewormers and improving overall animal health. In the swine industry, BLUP has been applied extensively to improve genetic resistance to viral and bacterial diseases such as porcine reproductive and respiratory syndrome, often referred to as PRS actinobacillus pleuropneumonia, and Escherichia coli scours. PRRs, often referred to as the AIDS of pigs, causes severe economic losses due to reproductive failure in sows and respiratory disease in piglets. BLUP-based genomic selection, or GBLUP, has allowed breeders to identify genetic markers associated with PRRS resistance, leading to healthier and more productive pig herds. Furthermore, BLUP has improved resistance to post-weaning diarrhea, a leading cause of mortality in young piglets. Genetic evaluations have demonstrated that selecting sows with favorable EBVs for immune response traits has reduced piglet mortality rates by up to 15% in commercial herds. The poultry industry has benefited significantly from BLUP-based selection for disease resistance traits. Modern poultry breeding programs incorporate BLUP to enhance resistance to bacterial, viral, and parasitic diseases, including Newcastle disease, a major viral disease affecting global poultry populations, infectious bronchitis, a respiratory disease impacting egg production, Marek's disease, a deadly viral infection in chickens, and coccidiosis, a parasitic disease causing significant mortality. By incorporating genetic evaluations using BLUP, breeding companies have successfully developed disease-resistant broiler and layer strains, reducing the need for antibiotics and vaccines. Studies have shown that BLUP-based selection can reduce Marek's disease mortality by 40 to 50% over multiple generations, improving flock health and productivity. As climate change intensifies, heat stress, drought conditions, and extreme weather events pose significant challenges to global livestock production. BLUP has become a key tool in selecting animals that are better adapted to extreme environmental conditions while maintaining productivity. Heat stress is a major concern for dairy and beef cattle, particularly in tropical and subtropical regions. Elevated temperatures lead to reduced milk production, lower fertility rates, and increased mortality. BLUP-based evaluations have allowed breeders to select animals with improved thermoregulation, meaning lower body temperature under heat stress, higher milk yield persistence in hot climates, and increased sweating and respiration efficiency. For example, in Holstein cattle, BLUP-based selection has led to the development of heat-tolerant lines with a 10 to 15% reduction in milk yield loss under heat stress conditions. Similarly, in Brahman and crossbred beef cattle, genetic evaluations have improved adaptability to hot climates, enhancing growth and reproductive performance. Sheep and goats are vital livestock species in arid and semi-arid regions, where water and forage availability fluctuate significantly. BLUP-based selection has been applied to improve feed efficiency under limited pasture conditions, enhance water use efficiency in desert-adapted breeds, and increase resistance to metabolic disorders associated with drought stress. For instance, studies on Dorper sheep have shown that genetic selection using BLUP can improve drought resilience by up to 30%, ensuring consistent growth and reproduction in water-scarce environments. BLUP has also been integrated into aquaculture breeding programs, particularly in selecting fish and shrimp species that can tolerate disease outbreaks and environmental fluctuations. For example, Atlantic salmon breeding programs have used BLUP to enhance resistance to sea lice infestations. Tilapia genetic evaluations have improved resistance to Streptococcus infections and temperature fluctuations. Shrimp breeding programs have incorporated BLUP to select individuals resistant to white spot syndrome virus, a major threat to global shrimp farming. The future of BLUP-based genetic selection in disease resistance and environmental adaptation will be shaped by the integration of genomic data, or GBLUP. The incorporation of whole genome sequencing data into BLUP models is expected to enhance prediction accuracy for resistance traits. Machine learning and AI applications, the combination of BLUP with deep learning models, will refine the ability to predict complex disease resistance patterns. Advanced BLUP models will incorporate multiple environmental and genetic factors over time, improving selection for resilience traits. Precision livestock farming, the use of real-time sensor data to complement BLUP-based genetic evaluations, will allow for more dynamic and responsive selection strategies. The future of BLUP in genomic selection is expected to be shaped by advancements in high-throughput phenotyping machine learning integration and multi-omics data incorporation. The development of hybrid models that combine BLUP with deep learning and functional genomics will likely enhance prediction accuracy and accelerate breeding cycles. One of the ongoing challenges in BLUP-based genomic selection is the need for high-quality genotypic and phenotypic data. While genomic sequencing costs have declined, large-scale data collection remains a challenge in many breeding programs. Ensuring accurate phenotyping and maintaining diverse genetic resources will be essential for maximizing the potential of BLUP in future genomic selection efforts. Another challenge is the computational complexity of handling large genomic datasets in BLUP models. The development of more efficient algorithms and parallel computing strategies will be crucial for scaling BLUP-based genomic selection to larger populations and complex traits. Best Linear Unbiased Prediction, or BLUP, has revolutionized genetic evaluation and genomic selection, significantly enhancing breeding efficiency in livestock and crops. The integration of BLUP with genomic selection, known as GBLUP, has led to substantial improvements in prediction accuracy, genetic gain, and disease resistance. While alternative genomic prediction methods continue to evolve, BLUP remains a cornerstone of modern breeding programs due to its statistical rigor, computational efficiency, and practical applicability. The future of BLUP in genomic selection will be shaped by advancements in machine learning, functional genomics, and high-throughput phenotyping. Overcoming challenges related to data quality, computational demands, and environmental variability will be key to ensuring the continued success of BLUP-based genomic selection in breeding programs worldwide. Through continuous innovation, BLUP will remain a vital tool in addressing global challenges related to food security, climate resilience, and sustainable agriculture.